Hi everyone, wishing you have an amazing day today. So now we present to you our biology outreach. But before that, I would like to introduce you guys our group member. The first one is SHUHADA FATIHAH, AMIRA SOFEA, SYAFIKA ALIA, SHAHERA AFIFA, SYAFIAH IFFAH, and NURDAYANA. This is all about movement of substance across a plasma membrane and its application in daily life. So let's get started.
So I am going to tell you about the necessity of movement of substances across a plasma membrane. Did you guys know that living cells require substances from the external environment to carry out living process? And at the same time, the metabolic process in cell produce waste that need to be disposed of from the cell. All cells must allow some substances to move into and out of the cell to maintain the living process. The movement of substances into and out of the cell is regulated by the plasma membrane. Okay, so plasma membrane is both permeable and impermeable. A membrane is said to be permeable to a substance if the membrane allows the substance to pass through it freely. Then it will become impermeable if a substance is unable to move across it. Then it proves that the plasma membrane is a selectively permeable membrane. Okay, then what is a selectively permeable membrane? So basically, selectively permeable membrane only allows free movement of certain molecules across it. And it also prevent or limit the movement of other substances. Okay, so why is the plasma membrane has a selectively permeable property? The phospholipid bilayer and protein itself determine their own permeability towards specific substances.
Concept of movement involved by doing pickles is osmosis. What comes across your mind when we talk about osmosis? Osmosis is defined as movement of water molecules from high water potential to low water potential through a semi-permeable membrane. So now, let us focus on the diagram shown. On the left side, we have the high water potential, which is low concentration in salt. On the right side, we have low water potential but high in salt concentration. After some time, we see that the water molecule from high water potential diffuse to the other side. This is osmosis process. But how long will this movement take place? It will occur until concentration of water is balanced, which we call as dynamic equilibrium. These are important things you need to know about osmosis. The first one, it is a passive transport which is occur in water molecule only. It does not require any energy because it moves from the high water potential to low water potential, which is down the concentration gradient. Okay, now let's take a look at the pickles. Cucumber, which is a high in water potential, will diffuse out from the cell to the salt solution through what movement? Through osmosis. Osmosis will stop until both achieve equal concentration or known as dynamic equilibrium.
The cucumber is soaked in hypertonic solution. Sugar solution is hypertonic to the cell sap of the cucumber. Water diffuses out of the cell sap of cucumber by osmosis. The vacuole and the cytoplasm in cucumber will shrink, causing the plasma membrane to be pulled away from the cell wall. This process is known as plasmolysis. The cucumber becomes soft and can be bent.
First thing first, you need to prepare a cucumber, some sugar, and generous amount of vinegar and water. You can add some chili pepper if you want to. Then, you need to cut your cucumber and chili pepper into small pieces as desired. After that, pour some generous amount of vinegar and add about 2 to 3 tablespoons of sugar. Then, stir until the sugar dissolves and add some water to adjust the taste. Put the cucumber and pepper into a container or a jar. Pour the mixture of sugar and vinegar that we have made before in the container that contains the cucumber and pepper. Close the container with the lid tightly. Keep the pickles in the refrigerator and wait for a few hours to let the movement of substances across a plasma membrane occur. After a few hours, check the pickles if it has become turgid. Unfortunately, we have reached into the last session. So now, let's move on into the question.
Next, question part. So the question is, state precautions of this experiment. We looked at the task given, it is state. So, we don't need to explain, we just have to give the answer. The first one, the cucumber have to be cut equally. Secondly, volume of each ingredient, especially liquid, have to be the same. Last, cucumber and the container have to rinse first before use.
The next question is how to preserve eggs. So this is the ingredient that we need to preserve eggs. We need some salt, eggs, water, and a container. Keep in mind that we also need a stove to boil the water to dissolve the salt. First, wash the eggs and let it dry. So this step is important since hygiene comes first in order to remove some dirt and excess bacteria. Then, boil some water and mix it with salt. Stir until the salt dissolves and let it cool. This step is to make the concentrated salt solution that are hypertonic to the egg cell. After that, carefully put the eggs into a container. Eggs have a thin shell that can break anytime, so we need to handle it carefully. Then, close the lid firmly and store in a dark place for about 2 or 3 weeks. The salt solution will prevent the growth of bacteria in the container, and the eggs will last longer because there are no airflow and no sunlight.
In preparation of our assignment, we had to take the help and guidance of some respected persons who deserve our deepest gratitude. As the completion of this assignment gave us much pleasure, we would like to show our gratitude to our highly respected teacher, Puan Wan HASNIZA Binti Hassan, for giving us a good guidelines for assignment throughout numerous consultations. We would also like to expand our gratitude to all those who have directly and indirectly guided us in completing these assignments. So that's all from us. Thank you.